Okay, and I know that some of you are still uploading, and so the last time I cut off an upload in my other class, I had one student that just basically had a meltdown and was mad for the rest of the day. So now I'm not cutting them off anymore. Okay, so if you want to sit there and fool around with that, fine, I don't care. Don't miss what I'm saying. Okay, that's what I really want, is I want you to get what we're talking about. So, intermediate filaments, very different than what we already talked about because these are not polar. And Courtney, that means?
They do not have separate sides.
Different sides.
Different sides.
So that means the two ends are going to look the same. Okay? Now, they're made up of proteins. And how can proteins look the same? How is that possible? Well, it's the combination of proteins that get put together to do this. So they start with an individual monomer. Okay, a monomer is just a protein from the N-terminus to the C-terminus. It happens to have this structure. It's globular, and then it has sort of a long stalk and globular at the other end. So you start out with one, and that's clearly got different sides, yes? Mm-hmm. All right. Well, we put two of those together in the same direction. So now, right, the N-terminus and N-terminus, and these guys wind, and now you have a C-terminus. Clearly different sides still.
Yes.
Okay. Now this is where they become the same. We actually put together two dimers, but the dimers are put together anti-parallel. That means, so two going this way, two now going this way. So you have an N-terminus here and a C-terminus, and then voila, look what happens over here. We have N-termini and C-termini. So now the ends look the same. Does that make sense to everybody?
Yes.
That is what we call the tetramer. Four of them together is what we call the protofilament. Okay? Remember what the protofilament was in globular actin? How many was it?
2.
The protomer was just one string. Remember how I said to remember what the protomer was how many did you need to put together to make the final? 2. So those you needed one string twice. And what was the protomer in microtubules? The protofilament. There's one string of those. And you need 13 to put those together. You with me? Courtney.
Okay, you just kind of confused me. So there are not 13 protofilaments?
There are 13 protofilaments.
But
13 of them.
Okay.
One is considered the protofilament.
Okay.
And then you need 13 protofilaments to make the microtubule. In actin, you put together one string of globular actins, and you only need 2 of them to make the actin. Okay, here we have a tetramer. Okay, and how many total things did I say? I know you know the answer. So we need 32, but this already has 4. So how many of these do we need to make an intermediate filament? 8. So 8 times 4. Right? Is that good math? Is everybody clear on that? Actually, I was having a brain fart right there. I'm like, isn't that 24?
So it would be 8 protofilaments.
8 protofilaments, exactly. All right, and I don't think this picture helps you. So I think I put in another picture, didn't I? Yeah. Okay, so here's my silly little picture. Okay, if these guys weren't here, all right, you would say, here's 8, 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 here's 8. No, I'm lying. Here's 4. Shit. Fuck. Take that back. You know, once you record it, you can't really... 4, 4, 4, 4, 4, 4. But it's not just going to be one of these. Right? Think about globular actin. To make that protofilament, we put a globular actin, a globular actin, a globular actin, a globular actin to make a protofilament. So here we're going to make a tetramer linked to a tetramer linked to a tetramer. And so you have one string of these, but then you have 8 total strings. Does that make sense? as I'm trying to be a string. I said something had 13 not only the day before yesterday, but today. Which one has 13? It is microtubules. So is this ties cutting off blood flow? Yes, 13 protofilaments. Is that getting longer? It is. Wow. He's looking very professorial. Okay. So this structure, what I hope you get from this, what would you say now that we built this? What would you say? It's like a wall. So it's going to be really strong. Okay. There's lateral interactions, between these guys. And remember what we said about bricks. Notice the staggered structure.
Oh, yeah.
Oh, yeah. See, they're staggered, so it really does create this incredibly strong filament. So intermediate filaments are mixed. Okay. I do like this picture, except for it's right there on where somebody drew on the board, right from the very beginning. Okay, and so if I just blow that up a little bit, what is it you're seeing when you see these blue dots versus the white dots?
The N versus the C terminus.
Very good. So you have two N termini right here that are bound to two C termini, right? And so this is very different arrangement than what we saw on the microtubules. The microtubules had all of the betas on the top and all the alphas on the bottom. And on the microtubules, it was really a hollow tube, and this one's actually filled. So they're completely this is a big, thick rope. Okay? Okay. Yes?
The wall kind of gets rolled up.
Yeah. Not roll. I wouldn't say, right, it's just made. So it's more like it's all braided together. So in this EM picture, they're showing you, okay, if I show you this structure compared to this structure, which one is which? What is what?
The right intermediate.
On the left?
Intermediate. The bottom.
Okay, here's one with this arrowhead, and here's one with an arrow. So, which one is which? You know the answer to this. Go back to your chart. The bigger one is a microtubule, and this intermediate size one is
The intermediate.
an intermediate filament. Right? And then all the little tiny skinny ones might be actin. They might not be actin. It's hard to tell from this image.
How do you know that?
The only way you're going to know this is if somebody gives you something like this and says, you wouldn't know just looking at it. You'd have to have some knowledge about what you were looking at, right? I can't just give you a picture.
Why are microtubules called microtubules?
Why are microtubules called microtubules? Well, everything's micro. Yeah,
Aren't they bigger than?
they are the biggest.
Yeah.
That's not my fault. Not my fault. Do you want them to be the macrotubules? And then we have, what would we have? Macro,
Intermediate.
micro, and nano? Yeah, so they're coming, right? Somewhere down the line, and then they're gonna name them all wrong again. Mr. Muselli.
Are those in tendons and like ligaments too, or those just in cells?
So are those in tendons and ligaments,
Which are cells.
or are they just in cells? So he's turning red. I'm not really I'm not trying to embarrass you. So what are tendons and ligaments?
Cells.
Actually, tendons and ligaments aren't cells. I think they're aren't they extracellular matrix molecules?
Connective tissue.
They're connective tissue. See, so Mr. Muselli, I don't know the answer, to be honest with you, but every cell will have these in them. So that's not what you were asking. You're thinking of what's outside of cells.
Yeah.
you're thinking of stuff that connects.
Yeah.
So if that's a tissue, if it's tissue, literally a tissue, that means it has cells in it. But like your bone does your bone have cells? Is it all cells? A significant portion of it is not cells anymore. I mean, what is it?
The product of your cell.
It's the product that's released out of your cells, so it's calcified materials that originally came from cells. This is where my physiology would really, like if I had some, that's why I want to teach physiology one semester, so that I can relearn this stuff. I'm not sure I ever learned this answer, but will you teach me?
If I knew that,
Well, can you find out?
yeah.
That would be really great.
Alright.
Okay. But the answer to your question is no. Okay, this is not how things are connected. This is only internally inside of cells. When we talk about junctions, which is coming next, junctions allow cells to be connected cell to cell through an extracellular interaction. So we're getting there. Okay, an extracellular matrix are molecules that are secreted out of cells that also help connect things together. You know extracellular matrix molecules, you know collagen. Collagen is secreted from a cell, and collagen helps hold things together. You might have heard of elastin. It's another extracellular matrix molecule that helps hold things together. All right? But the tendons and ligaments that you're talking about, which are connected to a bone, okay, those are, I actually believe you now, they're cells. They have to be cells. What else could they be? I have a ringing in my ear. Anybody else? No? Great. Okay, so I don't do a ton of stuff on intermediate filaments because there's a million different intermediate filaments. All right, there are 5 flavors, 5 different kinds of intermediate filaments, and I pointed them here. So there are axonal intermediate filaments, epithelial filaments, vimentin-like filaments. That's a great name. So somebody found a protein called vimentin, and they said, oh, vimentin's an intermediate filament, but then there's all these vimentin-like filaments. Oh, great. Thank you very much. And nuclear filaments. Is that only 4?
Yeah.
I'm really sorry, Ben. Counting is hard for me. Okay? You got married once. Yeah.
Is Vimentin its own, and then Vimentin Life,
No, just vimentin-like.
and then Vimentin?
Yeah, it's one of them.
Okay.
Yeah. Okay, so I want to talk a little bit about the different filaments because you have heard of them, you just never knew what you were hearing about. And so you've heard of keratins. Yes?
Yeah.
Hair, nails, rhinoceros horn. Okay. Well, this is just like big names. In different tissues, they're going to have different names, and you don't want to know names. All right, trust me.
I don't.
All right. You've heard of another one. You've probably heard of the nuclear filaments, lamins. No? Nuclear lamina. Guess what's on the nuclear lamina? Lamins. Okay. So neurofilaments. In neurofilaments, right? This is a way better picture of a neuron than I've ever drawn. Right? In a neuron, you have dendrites that go to other axon terminals, right? And then you have the cell body where the nucleus is, and then you have an axon. And in the axon, you have neurofilaments. And the neurofilaments are there to help with structure. And the reason that you actually do know a little bit about this is because fibrosis, it's a good idea, okay, but that's not an intermediate filament disease. I'll tell you the one that is. How about Lou Gehrig? Does anybody know Lou Gehrig? No. Nobody know. Who was Lou Gehrig? Lou Gehrig. And what did he die of? Good. And does anybody know what the name of that is? ALS? Amyotrophic lateral sclerosis. It's a sclerosis. And does anybody know how they figured out what his problem was? He's a baseball player. What do baseball players do?
Baseball.
They play baseball. And what is it about their baseball that you could probably detect that they were having some issue?
Their swing.
Muscular problems. So he went into a batting slump. He was a very, very good batter. And all of a sudden he was having trouble hitting. And that's how they figured out that he had this disease. And so it causes this massive problem with muscle movement. Right? So how does a nerve end up causing, right? So this is a disease of the intermediate filament of nerves. How does that end up causing muscle problems? So all of your muscles are controlled by nerves, right? Your brain is sending a signal, right? So my brain is sending a signal to punch Stephanie in the head, right? So the signal goes from my brain to my muscle to not smack her, okay, to cut it short. And if she happens to do this, then I hit her, but you know. So, but your brain is controlling that muscle movement. And so what he had was this deficit in being able to control his muscle movement. And he died very young. Okay, this is a bad disease. There's no cure for it. We don't have really anything we can do for it. So a lot of these things we've only found out through people having problems, and then what is the problem, you know? How did that happen? So he had a neurofilament problem. Diseases associated with these neurofilaments. Sorry, I'm going to skip that for the... You all know about epithelial intermediate filaments. So the thing that holds your skin together are the keratins. And those keratins create the structure, right? So if you just have skin cells sitting next to one another, how hard do you think that would be to break them apart? Right? Not that hard, okay? So there's actually these filaments that help us. So you see on here, what are these green dots? They're junctions. Do you know what junction they are? The tight junctions. So all of your epithelial cells, your polarized cells, are held together by just a tight junction. But in addition to that, associated with the tight junctions are these filaments. So they're not showing you. There's green in this cell too, and there's green in this cell, but they're not showing you. Why are they not showing you? They're just trying to show you one in each. All of the purple is in all three, and the red is in all three. So you have all of these filaments in all of your cells, so it helps to hold them together. And when we do junctions, this will become more obvious. Of any, and Desmin, okay, Desmin, you might have heard of Desmin. Why? What are these guys?
Muscles.
These are muscles. So Desmin is one of these muscle intermediate filaments. It actually helps hold together the intermediate filaments of your muscle fibers. So the yellow here is the Desmin, and it's sort of holding on to the muscles, and it's actually holding on to a specific place in the muscle. We're going to talk more about this. The Z-disc. I don't know if you guys remember what muscles look like. All right, so you have, oh, I need a bigger. Bigger is better, right? So if I were to draw for you a muscle, this is the way most people draw muscles. And what am I drawing? This is a sarcomere. Do you remember the sarcomere? No, not really, but that's okay. So there were thick and thin places where it was dark and where it was light. Do you remember that part? Okay, so the green, what I'm drawing are what we call thick filaments. The red are what we call thin filaments. And guess what the red are going to be made out of? Actin. And so the area where it's got both of the filaments, So this part is what looks dark in an EM. And then this part, from here to here, right, is what's going to look light. And on the ends, these things are the Z discs. And the Z discs are intermediate filaments. We'll do more of that, okay? You'll get enough of this that you'll be like, okay, I'm done with that. Oh my God, these are not in any kind of order. They were in some sort of order when I had a brain. I think, let's go backwards. Sorry. All right. The lamins, this is showing you a membrane. Do you know what membrane this is if I tell you that this is the nucleus? What membrane is this? ER, right? And so the ER membrane, right, is surrounding the nucleus. This is a nuclear pore complex, which we haven't discussed in here yet, have we? I just did it in 107, so. Okay. And on the inside, what nobody really knows about is that we have these lamins that are sort of coating the internal surface of the nucleus. And intermediate filaments are all about structure. So they're actually allowing there to be a nucleus so that the ER doesn't just collapse in. And what I didn't know until I started teaching this was our DNA, our histones that wrap up our DNA, are actually attached to the lamins. So,
I thought that it was those spokes that were from the microtubules were the things that held it together.
nope. The microtubules don't have anything to do with holding together the nucleus. Okay? They hold together the Golgi. They hold in place the lysosomes, but nothing whatsoever to do with the nucleus. So the lamins are on the internal surface, and so that means on this internal surface, so this is the outside of the nucleus and this is the inside. What's chromatin? It's proteins plus DNA. Our DNA is covered in proteins. Do you know what the proteins are? Histones. All right. And so that links it to the nuclear lamina, which has the lamins on it. Okay? Want me to draw it?
Yes.
Yes. Here's the cell. Here's the nucleus, right? And we all know that's wrong. But if I were to draw it like that, right, I would say that there's red intermediate filaments, and you're going to know that those are lamins. There's multiple different lamins, A, B, C. And those lamins help to hold DNA through interactions with the histones and also the structure of the DNA. I'm sorry, structure of the nucleus. Oh, now see, I am recording and I am erasing. There is no rhyme or reason to this, people.
It's been updated, maybe.
What's that?
Maybe it's been updated.
Maybe it's been updated. That's a good one. I like that. Yes?
So does that mean when we're doing a digest, we're breaking down the lamins or the histones?
So that's a great question. When we break open cells, okay, when we break open cells in the lab, what cell are you breaking open?
Yeah, bacteria.
Bacteria.
This is not.
They don't have one. They
It's
don't
just
have-
not the same thing.
Oh,
Huh.
that's clever.
So that makes it way easier because they don't have what?
Histones.
Well, number one, they don't have a nucleus. And where is their DNA?
In the cytoplasm.
It's in the cytoplasm. It's really one of the reasons that it's very easy to work with bacteria. But let's say we were working with a eukaryotic cell. In general, is it easier or harder to break open a eukaryotic cell than a bacteria? Harder, really? Why? There's more stuff. Well, no, sorry. I don't know how to break that down to like, okay, you're wrong. So it's really difficult to break open a bacteria. They have a thick cell membrane and cell wall, right? Even gram-negative and gram-positives, right? Gram-positive has a peptidoglycan layer that's huge, whereas the gram-negatives have a small peptidoglycan layer. And does anybody know what peptidoglycans are made out of? Sugars. What kind of sugars? Nag and NAM. And what do you know about nag and NAM? Nicole Crumpacker is on a roll, okay? She knows nag and NAM. What's his nag? Acetyl glucosamine, or something like that. And NAM? Uramic. That one's hard. I can't do that one. So I think it's neurominic, something. Okay, but what's really interesting about peptidoglycans is that it's a string of sugars that are connected through a covalent bond with an amino acid. And so what it does is it creates this incredibly strong structure. And gram-negative versus gram-positive just tells you whether they have this really thick cell wall of peptidoglycan. If they have a really thick one, it holds on to the stain, so those are gram-positive. And the ones that don't have as thick, it washes out, the stain washes out really easily, and they're gram-negative. So they're really hard to break open. You can break open a eukaryotic cell by shaking it. Okay, mechanical force like this does nothing, okay, to a bacteria. Bacteria, you can stomp on them and they're fine. But eukaryotic cells, if you look at them cross-eyed, they'll just pop open. Okay? So it's a totally different process when we do this with eukaryotic cells, okay? All right. That was sort of the introduction, and I always bring this guy up because he's kind of like a role model for me. This guy's name is Johnny Kennedy. He lived in England, and you might have seen him. There was a movie called The Boy Whose Skin Fell Off. You can watch it on YouTube. It's a little downer, but it has some. It's a little downer because he does die, but it's his spirit that really is an amazing thing. So he was born with this disease, epidermolysis bullosa simplex. Okay, who wants to say that? I would like you to recognize those words, right? If you saw that on an exam, you're going to say, oh, that's the thing that Johnny Kennedy had. And what it is it's a defect in keratin. So he had a defect in his intermediate keratin. And what did I say the name of the movie was? The skin fell off. So basically, if you touch Johnny Kennedy's skin like that, it would just break open and bleed. Putting on his shirt caused bleeding. Everything caused bleeding. So can you imagine living that kind of life? You see his hands are wrapped up because it's virtually impossible to live your life without touching things. So they try to protect him. And it was really his attitude about it. He became a spokesperson for the disease. And he's kind of old in this picture. What else might you expect if you have this kind of disease about his stature? Would he be a big guy? He's actually small in stature. He's a pretty short guy. Growth, you could imagine, would cause problems. And he was a womanizer. That was the other thing that kind of cracked me up. So girls liked him. All right, girls liked people who are genuine and honest. And he knew he was going to die at some point, but he really was a spokesperson for this. Kind of a terrible way to live. And if you watch the movie, so I highly recommend you watch the movie. What is on the next slide? Is there any more about him? No, that's sad. So he went and he wanted to make sure that everything was taken care of at his funeral. So he went and got a casket. And they're talking about making this casket. It's a little tiny casket. It's kind of funny looking. And he says, I want you to put on there, he put on a number. I can't remember what the number was. What's the number? 57. He's like, I want people to sit at my funeral and say, what's that number mean? And the guy's like, well, what does it mean? He's like, nothing. So I kind of enjoy that about him. I mean, he just has this kind of lighthearted way of living his life. He ended up dying of cancer. And that seems like an odd way to die when you have this disease. But what happens is, when your cells have to continually do repair, they 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 can turn into something that's no longer a functional cell. And so they can become cancerous, and that's what he ended up dying of. Sean?
So he died of skin cancer?
I don't know if it was actually skin cancer or a different kind of cancer, but it was a cancer. Yeah, I mean, it's on YouTube. Like I said, it's a little. I cried, okay, but I'm a crier. I'm like a weak stick. So.
What's that movie called?
The boy whose skin fell off.
Oh.
So I wanted to put this up here because if you look at this, right, it's just a keratin gene. It's one gene, and literally a substitution of a proline for a leucine. So that's one amino acid substitution. And that's what causes this disease. This is one of the ways, right? There's other mutations, but this is one. What do you know about a proline for a leucine? What's a leucine good at? Being nonpolar. Who said that? Thank you, Mo. So what's a proline good at?
Being weird and shit.
It's not necessarily big. What'd you say, Mike?
And shit.
It makes bends and turns, right? What does a proline look like? Does anybody have any recollection of their amino acids?
It looks like a pea.
No, it looks like a
It looks like a pea, that's
Yeah,
why.
basically like a pea.
It looks like a pig. Interesting. So if I were to ask you to draw for me a proline, you could all do it, yes?
Yep.
How many of you could not do it? Just show me your hands. And that's fine. It's fine. Just be honest with me. Okay? I appreciate honesty. So let's draw a proline because I don't want you to forget what we're doing. All right? What we're talking about is how our body functions, and it comes down to things as silly as an amino acid. What should I start with?
NCC?
Well, okay, I'll start with NCC. Now what do you want me to put, Courtney?
The H carboxyl.
Let's do the C terminus.
Yeah, okay, the O.
Okay, let's do the N terminus. What should the N terminus have?
1H.
So that's the tricky part, okay?
It connects.
So let's not do the N terminus yet. Let's do what's this structure going to look like?
It connects with
It's down.
Okay, so this is going to connect to a CH2, to a CH2, there's 3 CH2s, right? Do I have it correct?
Yes.
So what's maybe? Okay, I'm pretty sure I have it correct. What am I missing? H where?
There's one in the carbon.
There should be 2 H's here.
And one.
And a plus, thank you. And 1 here. Okay? So it really is the simple fact that now you have this bond as compared to a leucine bond. So this can't rotate anymore, and that causes any place where you have a proline to have a little bit of a kink in the amino acid chain. That can kill you in a very devastating way if it's in the right place in a keratin molecule. Does that make you feel? I think you should feel disconcerted by that. Okay? That means that you, all of you, right, every time you replicate your DNA, you have to do it so well that you don't cause these mutations. Right? All day, every day. It's incredible that you are functional. Yes? This is just one gene. Okay? So this is the ALS, right? Lou Gehrig's disease. I already told you he died because of neurofilaments that were important for motor neurons. And the interesting part of this is they've now linked ALS to mutations in SOD. Anybody know what SOD is? Superoxide dismutase. That's right. Superoxide dismutase. The hell is that? What's a superoxide? What's an oxide? Do we like oxidation? How do proteins feel about oxidation? Well, how do you kill things? Okay, oxidatively, yeah. So when you get a cut, what do? You use hydrogen peroxide? And what is that? When you pour the hydrogen peroxide on a cut, what happens? You get bubbles. Those bubbles are causing an oxidation reaction. That oxidation reaction can break open cells. All right? So now I just said to you, app's gone free. I swear I turned on Do Not Disturb, but it could have been on a different device. All right, so I just sort of said to you that oxidizing is bad, yes, because it breaks open cells. What do we eat now? What's one of the things that people say, oh yeah, you need to eat this thing or you need to eat this thing because it has something in it? Antioxidants. That's exactly why we're trying to eat antioxidants, to protect ourselves from things that can oxidize us. So, superoxide dismutase takes oxygen radicals and makes them into something that's no longer noxious. Noxious. How do you say that word? And maybe we just shouldn't say it because I don't even know how to spell it. It makes them no longer harmful. There we go. You can always find another word. All right? So, it makes them safe for you. How's that? Michael, you're laughing at me? You guys have questions? You guys okay back there? Yeah, you sure? Yes?
The oxygen radical makes what safe?
So, the superoxide dismutase makes the oxygen radicals safe. So, if you have a defect in that, now what's happening?
Radicals all around.
Well, now you have radicals that are going to cause problems. And where did I say this? Where we thought the SOD functioned? What disease? We were talking about ALS. So what's really interesting about this, and what you need to start thinking about is, well, if you have ALS and it's a disease of motor neurons, then that means that the superoxide dismutase is only defective in those motor neurons. Why isn't it defective elsewhere? Well, right? I don't have an answer for you. Right? Some genes are turned on in some cells and not in others. So maybe it's a different isoform. Maybe it's not turned on in other cells. Okay? Some things are ubiquitous. The keratin that I just said to you, right? You have keratin in a lot of different places. You would expect Johnny Kennedy's hair might also be defective. His nails might not be so good. Anywhere where he used keratin. So think about that when we talk about mutations in genes. How come it's not affecting every single cell in your body? Maybe it's only good for a particular circumstance. Okay, I hate to do this, but now we're going to do the hard part. How do you feel about that? That was the little introduction to intermediate filaments. We haven't done anything hard. That was just what they look like, how they function. Ta-da! Okay. I already said to you, polymerized, depolymerized. You're making, you're breaking. Okay, this is where it gets hard. There's a plus end, and there's a minus end. Which filaments are we talking about, by the way?
The polar ones.
Which ones are the polar ones? Actins and microtubules, not intermediate filaments anymore, okay? That was the end of intermediate filaments for the purpose of what we're doing now. All right? And the positive end, okay, this is the way I like to think of it. The positive end does everything faster. The negative end does everything slower. So, when I say polymerization, I'm basically saying we're making, and so polymerizing, you can polymerize on both ends of an actin filament. But the plus end is going to make faster than the minus end. So you can be adding subunits, right? So if this is the plus end and this is the minus end, okay, I'll start like this. Okay, you can add while you're just adding. Add. It's hard to do this. Add. Okay? You try it, okay? You try to be an actin filament. Not so simple. So we're going to see that we can grow them really quickly, and we can also depolymerize them quickly. And so then when we start taking them off, you can take them off faster from the plus end, so they can come off faster. Okay? So you want to? Okay, well, that's the best I can do. All right. So maybe we should start with, how do they start in the first place? So how do you start a filament in the first place? And it's called nucleation. What do you need to start making a filament? What do you need for actin?
Amino acids.
Oh, G-actin, thank you. You do. Mr. Buselli, we need amino acids to make G-actin, right?
Yeah.
Why?
Because of proteins.
Because it's just a protein. So we have this globular actin, but you need some subunits. Right? If you have one subunit in this whole area, can you make an actin? No. What if you have 10 subunits in this whole area?
It'll be slow.
It's going to be pretty fucking slow, people. Okay, what if you have 1,000 subunits in this area?
It's a little faster.
It's way faster. Things are going to bump into each other. So you're going to see nucleation depends on, it's concentration dependent. Polymerization is also concentration dependent, how many subunits are available. And depolymerization has a fancy name, which I actually love. It's called catastrophe. Okay, that makes sense, right? You're making a filament, and then all of a sudden, you're breaking the whole thing down. Gone. Catastrophe. So, these kinds of graphs, I'd like you to try to understand and be able to answer questions about them, like on an exam, which happens to be coming next Thursday. I did it before spring break, so you didn't have to do a shitload of stuff over spring break.
Can we move
The
it?
day before.
We cannot move it, no. It'll be okay, people. How many people are in genetics? Okay, so notice where all the genetics people are sitting, excluding Caroline. What the hell are you doing sitting in the back? Okay. They're all sitting in the front, so the overachievers are sitting up here. They're taking 360 and 380 at the same time, which we absolutely do not recommend. They all have Dr. Malone. Do you all have Dr. Malone?
Yes.
Did anybody cry after the first exam? Yes, we have one. Okay, two. We have two. So, actually, I was surprised there wasn't more crying in the room. There's normally a lot of crying because students don't, not you guys necessarily, but in 360, students don't believe it when she says you need to do the problems. So you just need to do the problems, and that means not do like one set of problems. You do every set of problems that there ever existed, so that when you come into the exam, you already know how to do the problem. It doesn't take you any time. The exams are specifically made to see if you actually just know how to do it. So if you don't know how to do it and you're trying to figure it out, you don't finish. Okay. How many people need to take 360? Okay, there was the little spiel. So you all now know the information. They're going to tell you, right? They're going to say that to you, and you're like, do all the fucking problems. Over and over and over. Like it's the back of your head. You need to know the ratios. Right? What's the ratios? 9, 3, 3 to 1. Okay? All right. If you look at it, I think cell biology is way easier.
Yeah.
It's not problem solving, except it should be. It's just more information. Okay, look at this chart. What's on the chart? What are we talking about? So this is looking time after salt addition, which doesn't really make a lot of sense. What's on the y-axis?
The percent of the subunits, the amount of subunits.
So this is basically saying, right, how many subunits do you need to really have a filament? So down here at low, right, you don't have very many subunits around, you don't have a filament. And the reason they're doing this with after salt addition, you need to cause something to happen. Okay? These subunits are all sitting here. They're waiting for something to happen, and something changes that says polymerize. Okay, whatever that is, right, in this case we can do it in the lab by adding a little bit of salt. So you have some subunits. The more subunits, eventually they're going to bump into each other. And then at some point, so they call this the nucleation phase because you don't have a filament yet, right? You just have some subunits. Okay. After a certain amount of time, okay, we call that growth, and now you have a filament. And then when it gets to the point where it's long enough, all right, then we do the adding and removing from both ends. Really what I want you to know is nucleation is when we're making filaments. It's all about having enough monomers to bump into one another. So when they're making this, how many protofilaments are here? Mrs. Acton?
1.
2. So they have to make, right, so up here it's a little easier to see that you have 2 strings wound around each other. So they're making 2 protofilaments at the same time. Okay, and this thing is going to become important. CC. So the CC is called the critical concentration. So at some concentration, you're going to sort of be at steady state where the filament isn't getting any longer or any shorter. Right? You might be adding and removing. So you might add one here and remove one here, add one here, remove one here. So it's not getting any bigger. And that's when we call it the critical concentration. So it's like where it's the tipping point. Above that concentration, you're going to be adding. Below that concentration, you're going to be removing. But it's a little more complicated than that. Okay. So the CC is where it's equal. But the complication is that the plus end has a different critical concentration than the minus end. Brian?
So at the CC, would you consider that treadmilling?
So give me 1 minute, we'll get to treadmilling. Okay. Because there are 2 different CCs. All right? So because there's different critical concentrations, that does something to it. Okay? Treadmilling is this, what do you do on the treadmill? You run. Do you get anywhere? You're in the same spot. What a stupid activity. Okay, let me just say, what are you doing? You're burning a lot of energy. You're not going anywhere. But that's designed for that, right? Sometimes what happens in your cell is the filaments are sitting there sort of in a treadmill. They're waiting for something to happen, but they're continuously changing. And there's a reason for that. You'll see in a minute. You're basically keeping the filament fresh. It's always new. It's always becoming new. And so in this picture, what they're showing you is, right, they're adding the red ones. Is this true? The dark red. And they're removing some of them, but they're adding and removing at the same rate from both ends. So what it means is you get new filament made, right? And the new ones, so these should be red too, right? But they're just trying to show you that this part of the filament is changing. It actually will be put in and then it'll come back out. It's hard for students to visualize this. What? For what purpose? So when monomers, when globular actin is added, it always is in the ATP form. And they call that the T form. Okay, so if we're talking about actin, right, actin binds ATP. And does it hydrolyze it? Does it get hydrolyzed? Yes, it does get hydrolyzed, Dr. Stein. That's what I meant to say. It gets hydrolyzed. So over time, as it's in the filament, it'll get turned into ADP. Now, can ADP get phosphorylated when it's bound to globular actin? Absolutely not. We don't have a mechanism for that, right? So think of these as getting older. So the longer they're in the filament, the more likely they're in the D form. What is the difference between the T form and the D form of globular actin?
ATP.
The amount of bonds.
It's just a difference in, it's not about energy for this. It's a difference in conformation. So the conformation, so the D form is less stable than the T form. So the longer it's around, the more likely it is to come back off. Okay? Once again, why do we care? We want to keep things fresh. So you're constantly, sorry, I have that. So we're constantly changing the filament, even though it's still there. Sometimes you don't want it to just go away. Catastrophe means it's gone. If we did catastrophe of all the microtubules in the cell, what would happen? What would happen to the Golgi? It actually completely disappears. It goes back, it gets sucked back into the ER. All right? You lose the Golgi altogether. We can do that in the lab. We have a drug that we treat cells with that causes it to just disappear. So I'm not going to ATP, versus whether it's in the T form or the G form. So this is just talking about, this is actin, and which one is changing in the tubulins? So we have beta tubulin and alpha tubulin. The beta cycles between GTP or GDP. The alpha stays GTP. Okay. So this is the D form leads towards disassembly. It's less stable. Maybe this will help. I have like 6 slides to make this maybe help. So now they're showing you green squares versus green circles. Okay? This is being made, so you're making a filament, and they're all, when you're making it, they're all in the GTP or ATP form. Okay? Somehow this one, right, got in there and it immediately got hydrolyzed. But most of the filament is in the T form, and as we add, right, if we add and we remove, right, so if you're adding to both ends, then it means you're going to have T's on both ends, right? And then it's harder for it to break down. But what happens, so treadmilling, Brian, is when you're adding to one end and you're removing from the other at the same speed. All right? And so what happens is the T's come on here and you have the D's here, and the D's are more likely to come back off. So it just keeps the filament sort of fresh and moving. Okay, this is harder things that we do in here. It's almost math, yes? It's graphs. So this is the one that did it for me. This is the way I understand this. Okay, let's just look at the dark green filament. Here's the dark green. Okay, and they're saying, all right, this particular filament is for the plus end. The plus end is going to have a different rate than the minus end. Okay? How is rate measured on a line like this? Slope. Slope. The change in y over rise over, what is it? Run. Okay, that's going back to, I think that's elementary school. I'm not really sure because that was a long time ago for me. Okay? So the slope here, you can see, is greater than the slope of the minus end. So the rate is faster on this end. Okay, now look at where the CC is. So the critical concentration for the plus end is below the critical concentration for the minus end. What does that mean? And we're talking about subunit concentration. So the amount of subunits around. Here we go back to that original example. We have a filament, and there's 10 subunits around. Okay, and let's say that's above the critical concentration for the plus end. Okay, so let's put a number here. Here's my 10. Am I above or below? I just lied to you, right? So I'm putting it below. Okay, what if I put it above? Let's make 10 here. Let's just cross this one out. Okay, so if I have 10 subunits, I'm above the critical concentration for the plus end, which means I can add to the plus end. Okay, but I'm below the critical concentration for the minus end. So what am I going to do from the minus end? I'm going to take off. So I'm adding, but I'm taking off. I'm adding, but I'm taking off. And as long as you're between these two, okay, that's called treadmilling. Okay? So we call that treadmilling because you're adding and removing. And there's probably, we draw it kind of large here, but it's probably smaller than that. We're just trying to show you that between those two concentrations, okay, you're going to be adding to the plus end but removing from this minus end. What happens if I then say there's 1,000 subunits? Right? I put 1,000 subunits in. Now I'm above the critical concentration for both, right? I'm way out here. So I'm above, if you're above the critical concentration, you add. So now I'm adding to the plus end and the minus end. So now I'm making a filament and I'm going as fast as I can. Polymerizing. And what happens down here when you have one subunit? You're below for both. You're breaking it down. Okay? So it's all about the critical concentration for the plus and the minus ends. How many of you are lost? And it's okay to be lost. Okay? Thank you for your honesty. That's really one. Okay? Mr. Muselli's not lost. You're a little lost? Okay. Are you lost in the big picture or in the nitty-gritty? Behind you.
Yeah, just like the middle part, I guess. I understand this.
When we have tons, okay, if you guys were all nucleating into a filament, and whatever that signal was to say go, all right? If so many of you around were above the critical concentration for both the plus and the minus end. Okay, so normally what I do is I just grab people up here from the front row. Yeah, let me have a group of people. Let me have. Come on. Let's be globular acting.
How many people do you want?
Like the whole front row. That means you guys too. Okay. So if we're all hanging out, you got to start somewhere, right? Okay. This is hard. This is a hard demonstration to do. Okay. Let's say you're the plus end and you're the minus end on this end. Okay. So if you're all attached to one another, so just hold hands for the moment. I know it's all cheesy. Okay. We need some of those wipes. Okay. And let's say there's no subunits around. Okay, what's going to happen to these guys? The only thing that really can happen is you're going to start losing from both ends, and you're going to lose faster from this end. Okay, so you're going to lose like this over there, and you're going to lose like this over here. Okay, so that's hard to do. Yeah, don't sit down. Now we're going to start adding you. Oh, wait, you want to just sit down and then be ready to stand up. Maybe let's have 4 people to start. 4. Okay? Thank you for my 4 middle people, and just get yourselves ready. Okay, so now, if everybody's available, right, we're going to add as fast as we can. Okay, but you're still going to add faster on this end than you do on this end. So you add on the click. Okay. What happened to my fucking globular actin right there? Okay, so do you see that it's slow side than it is on the other? Okay, now, here's the hard part. Half of you sit down. Okay, and this is gonna be hard because we're gonna have to, extra people are gonna have to do stuff. Okay? So now, if we're treadmilling, we're gonna add and remove. So I'm sorry, you're fine over here. This side, we're gonna need extra people. So we're gonna remove from this end when I snap, okay? But we're gonna add from this end when I snap. So, faster people. You see how the filament, go! It's supposed to be,
Yes.
it's a simultaneous, and so the filament is supposed to, it's hard to do with people, okay? So the filament is supposed to stay the same length, and that's what we call treadmilling.
The ones that come off,
The ones
do
that
they
come
come
on,
around and come back on the other side of it?
absolutely.
So then we could have just went to the other side.
If you're still in the T form, you can immediately get back on. If you're in the D form, you have to get, right, you have to kick off that ADP and get another ATP. Mr. Baselli.
If it's in the treadmill form and the plastic is added faster, wouldn't it grow?
It is growing. It's growing at that end, but it's coming off. So there's a place where it's equal rates.
Isn't the plastic faster than that? So would it get bigger?
So between those two concentrations, there's probably a smaller window than what I'm showing you, where the rate is the same. That's treadmilling. Okay? It's just a rate thing. This is just a rate. Okay? Yes.
I understand.
Thank you for playing the game, people.
Just the T to D.
So the longer you're in the filament, the more likely you're going to be hydrolyzed. Right? So that at some point, if a filament is around, right, let's say we really quickly polymerize a filament, and all of those are T. Well, then we start depolymerizing. Okay, they might still be T's on the ends, but the longer it takes to depolymerize, right, you're probably a D. And then all of a sudden, that's why catastrophe happens, because you end up at a place where you have all D's, and then it just goes, and it's gone.
Can you put a big picture for me? So this is inside each cell.
This is inside each cell. This is happening to your microtubules and your actin. Right? This is happening all day, every day. How do you think the spindle complex forms? Right? If you think about the spindle complex is making these spindle fibers. What happens to the spindle fibers? What are they attached to? The centromere. Right? They attach to the centromere at a place called the kinetochore. Okay, so they're attaching, and then what happens?
They're growing.
Well, they're not growing anymore.
They
They pull apart, and they're not like I can't make my arm shorter. Okay, right? They're not pulling like that. They're depolymerizing, and so they're holding on, and then it's just depolymerizing, so they're getting shorter. Okay.
Okay.
At the beginning, they're throwing these arms out. And we're going to talk about, there's three kinds of filaments. Okay, we only ever talk about the ones that attach to the kinetochore. But there are additional ones that overlap one another that hold the complex in place. Cleverly, they're called overlap microtubules. Okay, and then there's actually microtubules that stick out from the other side and attach to the plasma membrane. And they hold the spindle complex as far to the edge of the cell as it can, so that you get separation. Okay? Like I said, one of the hardest topics, these are all videos. I'll put a bunch of videos up on the Moodle site for you so that you can watch it, and maybe that'll help sort of cement it into your brain. Look what I have. I have a chart. So, how about you check your chart and see what did you get right and what did you get wrong? And can you fill in the rest of the chart? Right? So for microtubules, right, we had alpha and beta tubulins, and they bind to GTP with the beta cycling. Okay? What does this say? Okay. Preferential site of incorporation is obviously going to be the plus end. It's going to be the plus. I don't know why they put that in here. It's always the plus end. Okay? There is no end in the intermediate filaments, right? They're the same. Polarity, yes. No, yes. Enzymatic activity. So that means that this one's hydrolyzing GTP, whereas that one's hydrolyzing ATP. We're going to talk about motor proteins. This one has 2 motors. That one only has 1. Intermediate filaments, what are they doing? Structure, structure, structure. Okay. I don't talk a lot about this. Major group of associated proteins. You're going to see, so MAPs, MAPs, microtubule associated proteins. There's tons of these. All right. And I don't talk about plaquins, and I really don't talk about this. There's only so much we can really do in a class this size. Okay. Structure, right? Hollow. It says stiff, but I don't like that because it can bend, right? It's part of the stiff, whatever, hollow tube. This one's rope-like. This one's very thin and flexible. Okay, I don't care about the size. Okay, notice eukaryotes, intermediate filaments, animals. So over time, right, these have been changing, and the primary function. You should be able to recapitulate. What does recapitulate mean? Copy and paste. Did you really say copy and paste? No! Copy and paste is not learning. Okay? Copy and paste is copy and paste. It should come out of your brain. You should be able to do this. You should know this. You should be able to make this chart from scratch. Right? I mean, maybe I would have to give you some hints. Like I could give you some little parts. But then you could just fill in the parts that are missing, right? Okay? We're going to continue this discussion, and it gets a little more complicated because we're going to talk about the drugs that can affect filaments. And why do we care, right? We're kind of getting back to the big picture. Like, so why do we care? Well, one of the drugs that's currently used to treat cancer patients is a drug that inhibits microtubules. Why would that be good for a cancer patient? Stop cancer cells from doing what? Dividing. And what do you know about cancer cells dividing? They don't stop, and sometimes they divide faster. So we treat you with this drug, okay, that stops fast dividing cells. Well, what does that do for the rest of your cells? It stops them from dividing too. These chemotherapies are designed, right, to kill the cancer without killing you. Sometimes we kill you. Okay? It's not perfect. People that are on chemotherapy often die. And it's because cancer cells are you. They're not something totally different. They're your cells, and we're trying to target them in a way that, right, what's different about them? So this drug is called Taxol, and they almost always give it to breast cancer patients. It's one of the things because, right, these cells, once they get out, like if you have a breast cancer, and if it gets out, it gets into your lymphatic system. The first thing they check are your lymph nodes. They take a string of lymph nodes, and they check to see, are there cancer cells in that lymph node? If you have cancer cells in that lymph node, sadly, you are going to die at some point from that cancer. Because if it's in your lymph node, it's already gotten somewhere else. All right? So they treat you with Taxol with the idea that, okay, maybe we can grab some of those cells that are quickly dividing before they get too far. Oh, fuck, I'm supposed to be somewhere. Okay? So, see you next week.